I'm Chris Everist, and this is my brother Gary. Gary is a high school student back in Huntsville, and today I'm going to teach him all about protein synthesis. So Gary, basically, we're going to start. Protein synthesis is taking the DNA.
I got DNA.
Yes, you have DNA.
We have some of the same.
We have the same.
We're brothers.
DNA. We have brothers. High five, Gary. High five, Gary. So, basically, you're taking the DNA from the nucleus, and you're altering it and transforming it in certain ways, and we're going to transfer it out of the nucleus into the cytoplasm, and we're going to make protein out of it.
So this is the nucleus?
The nucleus is this
oval
here, yes, and this entire sheet of Bristol board is going to represent our whole cytoplasm.
Like a cell.
Like a cell.
Okay.
Like a cell. You learned that in grade 9 science.
You got it.
Okay, so your DNA is composed up
I have some of that.
Yes, you do. We have that. We share it.
Okay.
DNA is composed up of 4 different bases. Adenine, which is going to be represented by red. Guanine, which is represented by blue. Thymine, which is represented from black. And cytosine, which is represented from white. You'll notice that the DNA bases are paired together. A-adenine is paired with T-thymine. And adenine is a 2-ring base, and we call it a purine. And thymine is a 1-ring base, and we call it a pure pyrimidine.
So that's why that one's bigger.
Yes, because it's 2-ring, and this one is only 1 ring. So that's why this is bigger and that is smaller. So basically, we're going to pair this DNA together, see if you gather the concept. So A's are always paired with T's, and G's are always paired with C's, and we've represented, see how I've written it down here, how we represented A with red, G with blue, T with black, and C with white. So we're going to mix these up because they're all hunky-dory there, and we're going to pair this together, me and you together. So
As
red,
brothers.
as brothers, as brothers. So red is A, and it's paired with T, which is
Black.
black. So we're going to take a black, we're going to boom, 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 we're going to pair it together.
Okay.
And we're going to now, the next one is
Black.
black.
So we're doing red.
Red. Getting the concept already.
Huh?
White is a C, and C's are paired with
G.
G, as you see on the paper. So we're throwing a blue. Next one, black paired with red, which is adenine. So tossing that in there. Get the concept, how it's paired together. That goes there. That goes there. And there our DNA is paired together. Now DNA does not look like this. It is a double helix structure, and it is in a spiral.
All twisted up.
All twisted up. Double helix, and it's twisted. Does that make sense? So this
Sure.
would be
It's like a spring.
like a spring, exactly.
All right.
Exactly. All right, Gary. So the next step in protein synthesis is unzipping the DNA strand and duplicating it. So let's start by unzipping it. So comes through, unzips it. And what it's going to do is it's going to duplicate one side of it. So we'll duplicate this left side. Now the only difference it's an exact replica, except for the T's are replaced with U's, which is uracil. So we're going to replace our blacks with greens in our new strand. So this comes through, and in the nucleus, a new strand is made. Exact replica, except for the T's are replaced with U's. So that's happened, and then the DNA is going to zip back up. It's going to keep going. The DNA strand it goes on for forever, not forever, but for a long time. And so it's basically a bubble, and it just goes along, and it keeps duplicating this DNA strand.
Just making more.
Just making more. That's right.
All right.
So Gary, what's going to happen now is our RNA, our messenger RNA, is going to leave the nucleus through a nuclear pore in the nucleus, and it's going to enter the cytoplasm. Now it gets to the cytoplasm, he's going to find a ribosome. And our little hop character here is going to represent a ribosome. Okay, so the ribosome is looking for a start codon so it can start producing peptides. Now, the only start codon that is possible for it to start producing peptides is AUG. So, luckily enough, the very first three that we came up with is AUG. And so it reads it, gets the amino acids, and boom, out pops a peptide. And now it's going to continue to read it. It's going to find the next three. It's going to match it up. It's going to use the amino acids, and it's going to pop out another peptide. And now we're looking for a stop codon. Now it'll just basically stop codons. They trigger the amino acids to stop basically making peptides. And there's three different stop codons, UAA, UAG, and UGA. And the one that we have, UGA, that happens to be the very last three in our chain here. And so out pops one more peptide. And voila, we have ourselves a protein. Okay, now hang in, we're almost done here. So basically, we're going to recap what we've done in this section here for protein synthesis. So basically, we started with a DNA strand in a nucleus. We split it, unzipped it, we made a duplicate of one strand of the DNA, left the nucleus, went out into the cytoplasm, found a ribosome, put some amino acids and a little magic, out pop some proteins, and basically we got a tripeptide, which is a protein. It's going to go out into our body, and we're going to use it as an enzyme in some shape or form. And it's going to turn into energy or muscle. Gary, I just want to thank you again for taking your time. I hope you've learned something. Go back to playing Legos or something.
Hey, can I have my Lego back when you're done?
I hope. Yeah, you can have your Lego back. This is Chris Everist again. Thanks again for tuning in and watching. We'll see you next time.